Okay, so now we'll move on to Streptococcus pneumoniae, which is also called pneumococcus. And it is a gram-positive extracellular opportunistic pathogen with varying degrees of carriage among the population. It's up to 65% of children will have strep pneumo in their noses. They're not sick, they're just carrying this bacteria. And overall, under 10% of adults, but actually much higher in schools and hospitals. So up to 50% in schools and hospitals. Particularly, so anybody that's around children often has a higher chance of carrying Streptococcus pneumoniae. And this is the single largest cause of pneumonia in children worldwide. It's a significant health problem for children, both in developed and in developing countries. It's a sort of a universal infection of children. So the life cycle of strep pneumoniae is a bit different than everything we've talked about. All of the other pathogens we've talked about are things that when you come in contact with them, they either infect you or they don't. And if you get infected, it's a pathogenic infection. Or if they don't infect you, it means that you blocked it in some way, either at the mucosal surface or in a very rapid innate response. Strep you're living with a lot of the time. You know, a significant number of us on the call right now have strep in our nose and we're hopefully not sick. And so you've got the strep represented here by these little circles living in your nasal epithelium, hanging out in the mucus, like happy as a clam. You can transmit this bacteria to other people around you. This is why people that are around kids often have it, because the kids are transmitting it to them. And so what you're worried about in terms of causing disease is the spread from happy strep living in the mucus and not really doing much to invading tissues specifically. So if they invade the lung, and the lower lung in particular, then they cause pneumonia, which is why it's called pneumococcus. And then if they invade the blood, then it becomes bacteremia and it can become sepsis. And they can also invade the brain. It can become bacterial meningitis, or it can invade other surfaces. And it's one of the major causes, if not the major cause, of ear infections. And remember, when you were little, kids get ear infections all the time. And so we try to understand this conversion from carriage to invasion, and also the immune mechanisms that prevent spread and shedding to other individuals. So this is, again, happy carriage here. The bacteria actually have virulence factors, and different strains of bacteria carry different forms of virulence factors that will promote shedding and try to get to other people. And so pneumolysin is a protein that the bacteria make that allow them to shed more efficiently. And as it sheds, it actually causes inflammation. So inflammation can often be hijacked by a pathogen to use it to distribute more widely. I mentioned earlier, and I didn't emphasize it here, but sort of very different biologically and chemically, but sort of in the same way as TB, strep pneumo has this capsule around it, this instead of a fat, it's a glycocalyx. So it's sugar, and the different capsules will allow different amounts of shedding as well. Antibodies to that capsule can reduce shedding. So we've talked about the different classes of antibodies. IgG will actually block shedding and cause the bacteria to clump together, whereas IgA will not block shedding. And we'll talk about what IgA is good for in a few more slides. And then if you get a viral co-infection, that actually significantly promotes shedding, and it also promotes invasion. And so in that, I think, makes good sense, because you're really disrupting this epithelial surface. You're causing a lot of inflammation, and it just makes a happy environment for the bacteria to replicate. So that's the bacteria. Everything we've talked about so far has been on this sort of apical surface of the respiratory tract, where it's not really causing any trouble. And so when we start talking about invasion, we need to cross this respiratory epithelial barrier. Crossing the mucosal barrier, that's true of everything that we've talked about, is trying to break through a mucosal surface, the skin, the gut, the lung. any of these epithelial barriers. This is, again, another relatively large genome, and it has all of these short named proteins here are all bacterial virulence factors that are useful for driving the bacteria through the epithelial surface. It can do this in a number of different ways. It can promote endocytosis and then in turn promote exocytosis. So instead of staying in the cell, it just sort of takes the highway across. It can actually push itself through the tight junctions of the cells using various virulence factors to sort of push the cells apart. And different strains of the bacteria are going to have different virulence factors, and they're going to trade them amongst each other. This is what makes these pathogens really difficult to deal with long term, is they share genetic information very freely, and they use diverse mechanisms to try to overcome any of these barrier-based blockages. And that the same approaches can be used in the blood-brain barrier in the brain, and that's what's going to cause meningitis. Okay, so as I've said, we've mostly exclusively talked about type 1 responses, and now we're finally going to talk about type 3 or IL-17 based responses. The IL-17 response was only discovered in the sort of mid-2000s. And it took a little bit to then realize that it was really specific for these extracellular bacteria at mucosal surfaces. And as we sort of talked about with TB, there's this theme again of combined innate and adaptive responses that provide protection. So your first response is going to be to try to control the bacteria and block its invasion with traditional innate responses, which are going to be, in the case of these bacteria, both for strep and staph, the real key effector cell is going to be the neutrophil. And so this is a very pro-inflammatory innate immune effector cell that is phagocytic, so it can eat like a macrophage, but its primary function is not to be phagocytic. It's actually just to secrete lots of antimicrobial peptides and a lot of toxic chemicals like the ones that we were talking about for TB, but instead of secreting them inside their endosomes, they actually secrete them outside the cell. And so this is why they are very pro-inflammatory, because they dump all of this toxic material outside the cell to kill extracellular fungi and bacteria. So this is the solution the immune system has come up with. If you have a bacteria that doesn't go inside your cell and likes to colonize outside your cell, live in the mucus, you've got to get the weapons to the bacteria, and that's going to also now cause associated damage to other cells. So neutrophils, the neutrophil response is something that we tend to regulate very carefully, because if you have lots and lots of neutrophil responses, that's often what causes a lot of pathology in the lung. So what drives neutrophils to the site of inflammation? So they'll be called there by a lot of innate cytokines and innate signaling, but the most potent molecule that drives their trafficking is IL-17, which is going to be secreted by CD4 T cells in response to the microenvironment generated by these extracellular bacteria. So the IL-17 will act on epithelial cells to drive high production of these specific chemokines, CXCL1 and CXCL8, to bring neutrophils into the lungs to try to kill the bacteria. There are many drugs right now actually in development trying to block the signaling of CXCL1 and CXCL8 to limit the migration of neutrophils as a way of tamping down immunopathology. So in lots of lung infections, a lot of the damage that happens is thought to be caused by neutrophils. So if you can provide an antibiotic or an antiviral and get rid of the pathogen with medicine, then at the same time, you might want to shut down the host immune response because it's not needed anymore and it's now just causing trouble. And that's sort of represented down here, that you have tissue damage from the recruitment of these innate immune cells and these amplifying loops of signaling. So we made a lot of progress recently in terms of limiting pneumococcus. And again, it was the major cause of pneumonia in children. And one of the ways that has or one of the things that has limited the ability previously to make good vaccines and other interventions against strep pneumoniae is this capsule. And this capsule is, again, these polysaccharide sugar chains. And so the vaccine for these are actually capsule-based vaccines. So they are different strains of the bacteria, have different sugar structures, that they just put the sugars in different order, and they're slightly different sugar molecules. And so the vaccine is actually the sugar molecules cleaved off the bacteria and stitched onto a protein, because you don't want to give people necessarily the bacteria. And then this is called a glycoconjugate vaccine. So there are multiple pneumococcal vaccine platforms, and they're listed here: Prevnar 13, Vaxneuvance, Prevnar 20. The numbers refer to how many different types of capsules that they put in the vaccine, and they're generally recommended for kids or for the elderly, because again, these are the two populations that tend to have the biggest problems with these. And you're now starting to see actually people get recommended to get these even at younger ages, particularly if they have recurrent respiratory infections. So this has been an interesting thing epidemiologically. It's a great area for modeling, because there are, I believe, and sorry, I should have put this on the slide, 97 different capsules that have been defined for strep pneumo, and the highest valency we have in any of the vaccines is 20. So we're picking the top 20 and ignoring the other 77. So if you target population immunity against 20 of 97 capsules, what do you think is going to happen, even if you picked the top 20? So what happens is you see the reduction in the specific types of bacteria that you were vaccinated against, but then you just get new types that become the dominant strains. So the good thing is that there's still an overall reduction, and that's in the blue bars, of pneumococcal disease burden, because some of the top strains were really well-fit strains for infecting and invasion. But then you see this difference between the strains that are infecting people and the strains that are in the vaccines. So every few years, they actually reformulate the vaccine to shift to the now new top strains, while keeping some of the most dangerous strains previously in the vaccine. And so this is something that has to be updated constantly, and is sort of an ongoing monitoring. And so I mentioned before that secretory IgA didn't prevent transmission between people, but it does actually prevent invasion. And so those are also two things that can be divorced from each other. Is my ability to spread to you is different than my ability to protect myself from severe disease. And this is true across all the pathogens we've talked about. this is a particularly nice example of it. So IgA is a different class of antibody, and the way IgA is made is it's always a dimer of antibodies, and it's particularly enriched at mucosal surfaces. So you have a lot of IgA in your lungs, on your skin, and in your gut. And we mentioned IgA briefly in one of the earlier talks as one of the key regulators of the microbiome that you generate. And so IgA traverses the mucosal surfaces very, very easily and efficiently, and that's how you get very high concentrations, again, on the apical surface. So this is the part of the lung that's exposed to the air. And if you have high amounts of IgA, it will block the ability of the strep to be able to get across this membrane. Okay, so that was my summary of strep.